Well, I'm going to try and do something. A physician came to me and said, you know, there's a reason I'm a physician and not a molecular biologist because I really hate that stuff. Can you try and give me some information to help me to understand why you view things the way you do? So a little bit of this is an expansion of what I mentioned last year, as well as some new material. So first of all, you know, it kind of boils down to, I try not to be too much of a reductionist in life, but it seems like really there's sort of two broadly speaking perspectives on what Morgellons is. It's a psychiatric disorder, delusions of parasites, delusional infestation, something delusional, but there's not necessarily any organic biological component to it other than what could be said for any psychiatric disorder. Or it's a condition associated with one or more microbial agents, and behavioral or psychiatric symptoms are likely a consequence of the infection. So when I say microbial agents, well, viruses, fungi, filarial worms, arthropods, bacteria, those are the logical candidates. So this is sort of in a very simplistic way of looking at things, which actually in science has worked out fairly well for a lot of people. If Morgellons disease is associated with a microorganism or multiple microorganisms, there should be novel organisms found in samples from those with the Morgellons within the Morgellons population, but that are not commonly found in the unaffected population. You either have these or you don't. Have Morgellons or don't have Morgellons. And so that is sort of the approach I was taking. So I've done Western blots. I've done so many Western blots. I hate to think about it since about 1990. But I really like PCR because with the appropriate negative controls, it's not that difficult for what I've done to all throughout the last 20 years or so of doing research that to demonstrate that there isn't contamination. So we had already thought we knew that there were, well, we know from other papers there's Borrelia, but then we also thought we had identified Treponema, Bartonella, and H. pylori that Dr. Shoppe was just talking about. So those are the primers we use for PCR. Oops. And basically, PCR is got a small piece of it that is practically artwork and empirically derived, where you have to play with things over and over until you get it to work out right. And so there in the boxes, that's how many cycles had to be run for each of these primer sets to get an optimal signal that was not full of additional contamination of other things that start to get amplified that interfere with sequencing, as well as the temperatures and so forth. Now, I got burnt once, I don't know, what was that, 7, 8 years ago, by thinking there wasn't Borrelia present in Morgellons samples, but it was because my primers and the conditions just were not working. So now, everything we do, we try and find a positive control. So that if it's negative, we can run the positive control in the master mix, or separate, add the master mix to it to see if we can amplify it. So we purchased DNA for Borrelia from ATCC. That's a nonprofit that has mass quantities of various bacteria and DNA stored. We use DNA rather than the bacterium. Treponema denticola got DNA from it. H. pylori, they didn't have DNA, so we had to actually get the frozen cells and then extract the DNA from it. And then Bartonella, found someone out there in the universe who had some that was willing to just give it to us so we didn't have to go and dump $600 for it. And so when we did these, just to test the positive controls, I'm just showing you these to show you that there were bands. But the bands to me don't mean much of anything because I've gotten the right bands that were the wrong thing before. So what we did with each of these bands was cut them out, extract the DNA from the gels, and then sequence them. Here was the sequence for the Borrelia, Bartonella, H. pylori, and Treponema denticola. So now, here's the actual experiments. That was just to show that the primers work, we can amplify it, cut it out, sequence it, it's the correct thing. We have samples of punch biopsies and other samples that have been sent in. We extract the DNA, do PCR on that to see if we can amplify the DNA that we're interested in. We use agarose gel electrophoresis, then extract the DNA from the gel, sequence the DNA, and then use NCBI blast to see what the sequences are. So what I'm going to show you, every single one of these was sequenced. So this is not, well, maybe this is what this is. These are all definitely what we thought they were. So I have 8 samples that we ran through the entire process of looking for each of these different types of bacteria. So if you'll notice off to the left here, lesion 1 had Borrelia, Bartonella, H. pylori. It did not have Treponema present in it. The second lesion, and these are all from different individuals from different parts of the country, lesion 2 had Borrelia, Bartonella, H. pylori, and Treponema. So that particular Morgellons sample had all 4 of the bacteria that we were looking for. Off to the right, you can see where it says IDENT, that's the identities. So these are all 98 to 99% identical, the sequence with the published sequences. And also there are a couple of nucleotides that are consistently different from the positive controls, because we had we get one specific strain that was used for the positive controls. Well, there's always a variation. This isn't just a PCR artifact where it's a chance that they happen to be different, because it's different at that location every single time it gets PCR'd. So there's no question that these are what we were looking for. The third sample only had Bartonella, Helicobacter pylori, and Treponema. Anyway, these just listed, I put this compiled onto a table here where it's a little easier for you to see, but they're all 98 to 99% identical. And so here's the table. So sample 1, you can see, did not have Treponema. Sample 2 had all of them. So sample 2 and 6 had all 4 of the species present in it. Now, we did find some that did not have Borrelia. Samples 3 and 4. 3 had the other 3 bacteria. Sample 4 only had Bartonella and H. pylori. For these that didn't have it, they were redone with negative and positive controls to test the primers to make sure something didn't go wrong with it. And for samples 3 and 4, the controls amplified perfectly normally, and the controls were diluted down to almost 1 in 100,000, so there was very little DNA present, and yet we still got fairly decent bands show up from them. For whatever reason, these do not seem to have all of these. So for every one of the negatives for Treponema, H. pylori, and Bartonella down in that bottom one. So a couple of these only have 2 of them. Sample 4, as I said, Bartonella and H. pylori. Down at sample 8, only had Borrelia and H. pylori. Now, what does this mean? How do we interpret this? I'm going to come back to that. First of all, not all Morgellons is the same. I mean, even if individuals are talking to one another, some may have a little of all the symptoms, but 1 or 2 are particularly noticeable to them. So, I mean, when we say Morgellons, there's sort of a lumping of all the possible symptoms that a person might experience, and there are some more. Well, I had mentioned, and bear with me because there's a reason I'm showing you this. I had mentioned in the past that there seem to be 3 broad groupings of symptoms based on the OSU database that's been being collected, which wasn't working for like 9 months, and that was sad, and now it's working again. Notice I have the grayed out for fibers and joint pain. There's this entire block of people who have registered who don't have fibers. They feel they have Morgellons, they don't have fibers. Now, what was unique about a large percentage of that population was that the things that they mentioned the most as far as disturbing to them, itching, crawling, biting sensations, memory loss, difficulty concentrating, mood disorders. Well, coming from a neurobiology background, because the itching, biting, crawling sensations, anything that's going to affect sensory neurons could certainly cause those sorts of sensations, those are all neurological symptoms for the most part. Well, there's another group that has neurological symptoms, but does have fibers. But they segregate it out into these clusters. And for both of those, the without fibers and with fibers that I labeled neurological, joint pain was almost non-existent. There were 2 out of 35 that had joint pain. The rest of them listed it as completely absent. What could be different between these groups? And then there's a third group that I labeled the joint dysfunction group because that was the single biggest thing that on the registration page they listed as the thing that disturbed them the most. Sometimes elbows, sometimes knees, sometimes hips. Now, almost none of them also listed any comorbidities that were associated with joint pain. They have almost no of the sort of overtly neurological symptoms like the memory loss, difficulty concentration, the thing that is referred to as brain fog. There was not a one of them in this group that listed that as something that they experienced. Yes, it was kind of scattered. So the things I have in black were, yes, some of them reported it, some of them didn't report it. But 100% of this group, the joint pain was theirs. So now the reason I mentioned this was because last summer, I have a bunch of second-year medical students who are doing some literature searches for me. Basically, it's sort of like a work study for them, and it's during the May and the first part of June when they're studying to take their level 1 boards. And so what I do is I put them on one topic, and they're not together. They actually I don't even know if they know who is working for me because we never met together as a group. But I wanted them to do a literature search on the topics of chronic Lyme disease and post-Lyme syndrome without knowing anything. They'd all heard of Lyme disease, that was about it. So they had to come up with at least 2 review papers for each of them, as well as some primary literature. And basically, they were supposed to read through all this stuff and come back to me, tell me what they had read about, and come to a conclusion. Which one seems to have actually the best evidence? Well, just as an aside, 100% now for 2 years in a row have felt that chronic Lyme disease seemed to make much more sense to them, and the post-Lyme syndrome seemed a bit like a cop-out of, well, we can't figure out what's going wrong with you, so it must be something to do with what happened earlier. But when they finally do report back to me, I meet with them one-on-one, always Morgellons comes up. I mean, it just is a natural sort of a lead-in. So they ask some questions. Well, are there different species of Borrelia in these variant populations? And we know the answer to that now, in which I say, well, not from me, but apparently there are a lot of different Borrelias that could be involved. Are the varieties of associated bacteria different in the variant populations? And that's what I was showing you earlier. I don't know the answer to that, but it's a good question. They're obviously thinking about those. But then there were a couple of other questions that popped up. Do the bacteria cause the lesions, or are they somehow attracted to the lesions? And I thought, that's an interesting question. Well, they probably caused the lesion somehow, a massive inflammatory response, who knows what. But then the next question, are the same bacteria present in the lesions also found in the same patient's normal skin? I'm not often completely stumped or speechless when somebody asks me a question. And I just sat there for a minute and thought, that's a great question. I don't know the answer to that question. Well, so then I'm thinking, okay, so punch biopsies from lesions and unaffected skin from the same person. For me, that's a difficulty and a problem because my human subject's IRB exemption stipulates that all samples have to be de-identified when they go to the lab. When I had lab techs, if they were working on it, I de-identified them. If it's something I was going to work on, they de-identified it. So how am I going to do that? Well, okay, so this is a mea culpa here, all right? I spend about 30 to 50 hours a week working on the med school curriculum, and so I'm not in the lab very often. Well, guess what? That finally paid off because I found 2 matched sample sets that hadn't yet been de-identified, still in alcohol from, I don't know, 9 months ago or so. They might have actually still been in the boxes that they were shipped in. But at any rate, I was able to both de-identify and code them so that I knew they were from the same 2 people. So often with punch biopsies, what they do is take the margin, the border, some normal tissue, some abnormal tissue, so that it can be looked at from a pathological perspective. And for whatever reason, these physicians who took these samples also sent a sample of unaffected tissue. They didn't get these for me. What happened was basically they sent it to the pathology lab, and roughly half of the tissue was unused by the pathology lab. They had arranged for it to be sent to me. One from a right thigh, and sample 2 was from a right upper arm. I don't know what part of the arm it didn't stipulate. So same thing as before. I'm going to look for the same bacterial species, but this time within the same person from affected and unaffected tissues. And so here is the first person, and this sample came from New York. I'm allowed to say what states things came from without it being a problem. And so the lesions, the lesions had Borrelia, Bartonella, H. pylori, and Treponema. The non-lesion for the first sample only had Borrelia in it. And this is from the same person. The second person had Borrelia, Bartonella, and H. pylori, but no Treponema. The non-lesion only had Borrelia in it. And I was thinking, wow, this is great. So put onto a table so you can look at that just a little bit better. While it's true they didn't all have the same in the lesions, they had multiple bacterial forms found, but they were missing the co-bacteria. They only had Borrelia present. So I thought that's pretty intriguing. Well, as it turns out, a physician in the state of Washington sent parts of 4 punch biopsies, 2 from lesions from the person and 2 from areas with no visible lesions. So I just coded them as lesion 1, 2, non-lesion 1 and 2. Did the usual, extract DNA, PCR, and so forth. In lesion number 1, Borrelia, Bartonella, and H. pylori were present. In lesion number 2 from the very same person, only Bartonella and H. pylori were present. The non-lesions, the first non-lesion, L1, H. pylori, non-lesion area 2 had Bartonella and H. pylori in it. So putting that into a table, it's not quite as nice as what those first samples had. First of all, in the very same person, the two lesions did not have the same bacterial species present. And in the non-lesions, they didn't have the same bacteria present. You know, you have this fantasy world of, wow, maybe you can have Borrelia present all the time, and that's where the Lyme symptoms come from, but you have to have the others to make the Morgellons symptoms. Well, it's not going to be quite so clean as that. Look at lesion number 2 and non-lesion number 2. They're exactly the same. Now, would there be any reason why that might be, even if the idea that maybe you do have to have more than one type of bacteria present to cause the overt Morgellon symptoms from a non-lesion? I know of some physicians who have taken punch biopsies from areas where a patient will say, there's nothing there, but I feel it. There's going to be a lesion there. And so they will sometimes take a sample from that to do the pathology on it, which normally doesn't reveal much. I'm not saying that's happened here because I don't know where this came from, but it would be one possible reason that both would be present. It's just this is an area where a lesion is probably going to erupt from. So taking all of it together, there's some takeaways from here. You know, there were 3 samples. If you look up at the left now, this time it's for how many of each of those combinations as you read across the screen. 3 samples had all 4 bacterial forms present. 3 samples had everything but treponema. 4 samples did not have Borrelia out of the total. 2 samples only had Borrelia and Bartonella. 1 sample was missing H. pylori, but had the other 3, and so forth all the way down. And then even the 1 sample that only had H. pylori present. So what does that do with where I was trying to go with and look at this in that sort of simplistic way? Well, let's revisit the rationale. If Morgellons disease is associated with a microorganism or microorganisms, there should be novel organisms found in samples from those within the Morgellons population that are not commonly found in the unaffected population. Well, we haven't looked at that last part yet, but did we find some novel organisms that when you talk to most physicians go, wow, what are all those doing in there? Yes, 4 different bacterial species were found in Morgellons-associated tissue samples. Now, here's the thing. There was not one single Morgellons sample that was tested that had none of them. Now, did I have some that didn't amplify any of those? Yes, but then what I did was use Staphylococcus epidermidis primers, because that we find in every sample, normal and unaffected and affected. So it was like a positive control. Well, all of those that didn't amplify any of the 4 species that we think is somehow implicated in Morgellons also did not amplify the Staph epidermidis, which suggests the DNA was no good. It had been degraded. Something had gone wrong with it. So of all of those that we could amplify the Staphylococcus in, they all had 1 or more of those as I just showed you. So now, let's look at the flip side. It would need to not commonly be found in the unaffected population. See, that's way back when I started on this, I was just looking for it. This is such an unusual condition, there's got to be something different between the population that has Morgellons and those that don't. Well, let's look at the unaffected population. That was actually easier to get samples from pathology labs. Punch biopsies can be taken for various reasons, including looking at for cancer. So getting a bunch of those was actually not that difficult. And once again, Staphylococcus epidermidis was the positive control for it. So I had 7 different samples here that were all positive for, and actually I did more than just S. epidermidis. There were some other primer sets that were used just to try and make sure that it was working. They're not hidden in there. Not one of these 7 samples had any of those 4 bacteria present in it. None. Okay. If you're still with me, poor Mr. Darcy, he was sound asleep there. I was able to take that picture. My daughter's dog. But anyway, if you're not like Mr. Darcy and snoozing away unoblivious to the fact that someone's taking a picture of you, I want to go back to what I said at the beginning. There seem to be 2 differing views here. Well, in my research, since 100% of the Morgellons samples and 0% of the unaffected population samples had one or more of the specific novel bacteria, I have to reject one of those 2 basic ideas. I'm not going to call it a hypothesis because those aren't worded as hypotheses, but still, because Morgellons is a condition that is associated with microbial agents. And any behavioral psychiatric symptoms are likely a consequence of either the infection or from the stressors associated with any chronic disease state, or both. So, it's not overly sophisticated, and I tried to have a couple simple questions to answer. And in my mind, I did, I'll flip through my 3 pages of references too, just to show that I had some references. And I want to address for just a moment this business of going public. And I understand that when you've been burnt once or twice, it's really easy to lose trust. And if you've been burnt 20 or 30 times, it's probably really easy to lose trust. But in my experience, there's more good people out there than there are truly evil people. And so, being a mainstream scientist and working on something that is decidedly non-mainstream was a little risky. And occasionally when I get some hate mail, they'll say things like, I'm going to contact your president or your dean, and do they know what you're working on? Well, President Shrum, who is both the president and then the dean of the College of Osteopathic Medicine, the vice provost and my associate dean of biomedical sciences, my department chair, oh yeah, they know. They're quite aware of it. And I show them some of the data periodically. I was called in once to have a conversation after the CDC paper came out. And after the conversation, across the board, they were all unanimously, okay, well, good luck, carry on. So I think there are times when it's actually kind of, worth going out on the limb, as it was. And then thank you to the Holman Foundation and Cindy Casey, Casey Holman, who's become a friend over the years. All those researching Morgellons disease, and my family, who've been very supportive in this, because actually I once said the whole thing about, I feel like I'm climbing out on the branch and I have the saw in my hand. And my wife, who also is an RN, said, well, maybe the branch is low and you won't fall too far. So thank you very much, and I'll talk to you later.